is Dr. Jochen Ditterich, who is a professor of neurobiology, physiology, and behavior, and a core faculty member at the Center for Neuroscience and co-director of the Center for Neuroengineering and Medicine. Dr. Ditterich's research focuses on understanding the neural mechanisms that link perception and action, particularly in decision-making processes. His work investigates how the brain processes sensory information and integrates it with other knowledge to make decisions about when and what actions to take. By combining behavioral studies, electrophysiology, and computational modeling, his research bridges the gap between experimental and theoretical approaches. His lab also pioneers neuroengineering methods, developing innovative technologies that enable neuroscientists to tackle complex research questions. He develops new tools to treat cognitive deficits resulting from Parkinson's disease, schizophrenia, and other neurologic or psychiatric disorders. So please join me in welcoming Jochen.
Thank you, Marie. Good afternoon, and thank you for joining us today. So I was originally trained as an electrical engineer, specializing in information processing in computers and other technical systems. But over time, I got more and more interested in this ultimate biological computer that we have up here. And I wanted to contribute to the understanding of how it actually works. So I transitioned into neuroscience, did basic neuroscience research for about 2 decades or so, and only very recently started then working at the intersection of both fields, an area of neuroengineering. Okay. So now engineering, right, is obviously rooted in mathematics, computation, models, but these things also play a really important role in modern neuroscience. So, for example, modern recording techniques allow us to record brain activity from up to 1,000 or so electrical contacts in parallel, which results in really big data sets, right? So you've heard about the problem of big data, and you obviously need clever algorithms and quite some processing power to be able to analyze those data. What I want to focus on today here is the role of computational models in neuroscience. So now I could have told you some stories about how important they are for fundamental neuroscience and understanding neural function, right? So for example, as you've heard, we do research in decision making, and understanding how the brain is able to convert available information into a particular action choice critically depends on being able to work with mathematical models. However, since we just heard from Dr. Earn what is currently possible in terms of treating Parkinson's disease with deep brain stimulation, right, and what will be expected to be possible very soon, I thought I would tell you a somewhat different story, and that is a story about a potentially larger role of computational models in translational applications, in particular therapeutic neuromodulation in the near future. Good, so now, right, we have heard that the basic or traditional approach to deep brain stimulation relies on taking an electrode, placing it at a location in the brain, and then applying a train of electrical pulses, typically at a high rate. And so what that essentially does is really interfering with or disrupting the information processing at the location where that electrode is placed. So this works in Parkinson's because there is an imbalance between how much activity there is in 2 different pathways, and the stimulation allows to reestablish that balance. But you can imagine that this is not an approach that will work universally. Right? So if you told an engineer, right, that you want to fix a system by systematically jamming it, they would look a little bit funny at you because that certainly wouldn't be their first choice. Right? So how does an engineer go about controlling a complex dynamical system? And is this something that we might be able to do with the brain as well? So let's take the example of autonomously flying an aircraft, right? What an engineer would do in that case is they would start with a mathematical model that describes the behavior of that system, right? And then they would start taking a large number of measurements, right? So they would measure the height, the airspeed, the heading direction, whatnot, right? And based on the mathematical model, that would allow them to estimate what the current state of that system is, right? And finally, they would take a controller, again, informed by that mathematical model, to decide how they have to act on that system to get it into a particular desired state, right? Let's say approach a particular airport and then initiate a safe landing there, right? So how would an approach like this look like if we tried to approach it to the brain? So the idea is the following, right? So let's assume that we are able to implant a large number of electrodes into the brain, which allows us to monitor ongoing neural activity, right? And based on that ongoing neural activity, we should be able to estimate what the current state of that system is, right? And then, again, using a controller, right, we should be able to find out how we have to act on that system to get it out of a disease-related state, right, into a state that resembles more how a healthy brain operates, right? Good, so now I mentioned to you that mathematical models, right, are absolutely critical for that approach. But how do we get it from, right? So when you're dealing with an engineered system like an airplane, this has been designed by humans, right? So they know exactly how that works and how it has been put together. So you have a model typically readily available. But that's not the case in the brain, right? So what could we do? Well, we could potentially monitor that ongoing activity for an extended period of time and try to learn or extract from that data what an appropriate model of that would look like. And this is an approach that an engineer typically refers to as system identification. So before I tell you a bit more about what type of models we use for this, let's talk a bit more about what we mean by estimating the current state of a cognitive system. Imagine that you're making a decision what car to buy next, right? You would probably start out in a somewhat undecided state. You would collect some information, right? Over time, you would probably develop a preference for a particular model, right? And at some point, you would have to commit to a particular choice, right? The idea now is that this change of your cognitive state should be reflected in neural activity, and that in principle, we should be able to decode what that state is from observing that neural activity. So let me give you an example. We trained animals to make decisions about visual stimuli that looked something like this. Okay, so what you're looking at is a cloud of flickering dots, and embedded into that flickering, there are simultaneously up to 3 directions of motion with various strengths. And so what the animal is trained to do is to make a decision about what the direction of the strongest component of motion in that stimulus actually is. And then once they basically know, they are trained to make a goal-directed eye movement to one of those 3 choice targets here, right? Basically telling us, we think that, right, this was the strongest direction. I can play that stimulus for you again, okay? And you can try whether you can find out what the strongest direction of motion in that case actually is. Let's see, if it allows me to do that. No, it doesn't. Okay, let's go back. Okay, let's do it this way. So see whether you can figure out. Let's go back twice. So hopefully then, yes. Anybody have any guesses what that strongest component might be? You think down left? Any other guesses? It's hard. Pointing over here, right? Okay, right actually would have been the correct answer, right? But you can already see that this is not an easy decision task to do. So in this particular example, as is indicated by the numbers, actually 30% of those dots were moving to the right, 10% top left, 10% bottom left, right? Okay, so now while the animals were performing that decision task, we recorded activity from a part of their brain that is called the parietal association cortex. And the particular recording technique that we used for that experiment allowed us to selectively record from specific neurons, and those were neurons that were carrying decision-related activity, and they were selective for one particular choice out of these options that you have available here. Right. Okay, and I can now show you what actually happens to that neural activity as the animal is making those decisions. So how that activity unfolds on individual trials, individual decisions. Okay. So what we are going to look at here is a plot where on one axis I will show you the amount of activity of a pool of neurons that is encoding one particular choice that the animal could make. And this is just counting the number of action potentials that those neurons fire in a particular time window, versus on the other axis, how much activity a competing pool of neurons encoding a different choice that the animal could make actually carries, right? And so I will first show you a couple of typical trials where the animal made one particular choice. Let's call that choice just choice A. And what you will see is that the neural activity will start in an undecided region over here, and it will gradually move into the region associated with that particular choice. So then I will show you a couple of typical trials where the animal made a different choice. Let's call that choice B. And what you will see is that over time, the neural activity will again start in the undecided region, but will then gradually move over in that region associated with choice B. And finally, I will show you two examples of particularly interesting trials. So these are trials where the animal initially favored making choice B, but didn't end up doing that, actually changed their minds, and then in the end moved over and actually decided to pick option A instead. Okay. So this is what it looks like. So these are the typical choice A trials, right? So we start in the undecided region. Oops, sorry, let's do that again. Up, sorry, here we go. Up, no, here we go. Okay, good. So we start in the undecided region, right? And over time, our neural activity moves into that region associated with choice A. These are the choice B trials. Again, we start in the undecided region, right? Over time, the neural activity will move over here. And now you get to see those change of mind trials, right? Where the activity initially moves over here, but then actually curves around and ends up in that region. And that is actually the choice that the animal made in the end. Okay, good. So now I told you that in this case, we were able to select particular neurons, right? In general, when we are just implanting a large number of electrodes in terms of a chronic electrode array, for example, we cannot select what neurons we will be able to record from. And what we have to do in that case is learn a model first, and then after that, we can use the model to decode the neural activity. And I can show you for the exact same data what that approach would look like. So the type of model that we learn is a so-called dynamical systems model. And this is a model that has predictive power in terms of how we expect the brain state to change over time in the near future. Okay, good. So we are in this case, I actually just learned a simple linear two-dimensional model for you because that allows me to easily visualize what's going on. But we can also learn more complex nonlinear high-dimensional models as well. So where we are in this so-called hidden state space now, so the location where we are still reflects the current brain state. However, it no longer directly reflects ongoing neural activity. It is something that has been inferred from that ongoing neural activity. And you can think of that as a compact description of a more complex neural phenomenon. So to provide an example, right, if somebody tells you an inflation index, you have an idea how prices develop, right? And for that, I don't have to tell you what the exact prices of individual goods are, right? Okay, so what else do we see here? Well, you see those arrows, right? Those arrows is something that we refer to as a vector field. And so what they describe is how we expect the state of the brain to evolve, right, within the next near future, right? So for example, if the brain at any given time would be in a state here, we would expect that state to shift in this direction, and the long arrows indicate that we would expect that change to be rather fast. Or if the brain state were somewhere up here, right, we would expect it to shift in this direction, again, relatively fast. Or if we are at somewhere in this region, right, where we have much shorter arrows, that would indicate that we would expect the change to be much slower than in those other regions. Okay. So now, having that model, right, we can do our decoding. So I can now show you how, on average, the brain state evolved. And the first comparison that we are going to make is by comparing two different situations. One of them is one where the animal watched a stimulus that mostly provided evidence for making one particular choice, I called it choice 1 here, versus watching stimuli that mostly supported a different choice, choice 2, okay? And the average associated brain state will be plotted in red and green respectively. So what you will see is that early on, right, the brain state evolves together. The animal doesn't know yet, right, what kind of stimulus is going to be shown, but as there is more and more useful information for making the decision, you can see that the brain state actually splits, right, and starts to move in different directions. I can also show you what happens when we look at the average brain state depending on which choice the animal makes. So in red, you will see activity related to making one choice. In green, activity related to making another choice. And what you see in this case is that the activity rate starts at roughly the same location, but then moves in opposite directions and separates more and more, right? And so we still have something like an undecided region over here, right? We have a region associated with one particular choice and another region over there associated with another choice. So now how does model-based control come into the picture, since that is in my title? Okay. So imagine that we are dealing with a dynamical system that looks something like this. And the green shaded area over here would indicate healthy brain states. The red shaded area there would indicate disease-related states. And let's assume that we are currently in a particular location that is indicated with that red dot. And what we would like to do is to move the brain from this state into the state marked with the green dot up here. So we could try to do that on the shortest possible path, right? But those long arrows here that are pointing against us actually indicate that that will probably find quite some resistance when we try to do that, right? Instead, what we could do is actually try to move the state along this path, and this is something that should be much easier to accomplish, okay? And so this requires knowledge of the dynamic structure of the system, and this is what we call model-based control. So now, the final step that you have to implement is that you have to figure out what kind of stimulation you have to apply to the brain in order to make it undergo that brain state, okay? And so how can you do that? So in this case, you can learn from stimulating the brain with a number of different random patterns and recording what the brain's response to that looks like, right? And this allows you to figure out what the appropriate patterns are that you need to apply to make that state change happen. All right, so what are some of the envisioned potential applications here? Good. So we could imagine that you could apply that in situations where patients have difficulty with decision making, for example, pathological impulsive decision making. Could imagine applying it to working memory deficits, right? Or also attention deficits. So now, is this something you will see in clinical practice tomorrow? No, there's quite some research that needs to be done. It needs to be validated in animal models first before you can go into testing it in patients. But I still wanted to give you a glimpse of what an alternative approach to brain stimulation could look like in the future. And so obviously this is something, right, a very interdisciplinary project that you cannot just do by yourself. So we've been working with Dr. Kong, a control engineer, also with Dr. Brandman, a neurosurgeon, right? And with that, I would like to thank you for your attention and would be happy to answer any questions you might have.
Animals, thank you. And they look at those, dots. What part of the brain are they using? And does it make a difference whether they are the propensity to the left brain versus the right brain?
Okay, so first of all, they use many, many parts of their brain in parallel. So it really is a large network of regions that interact with each other. And in fact, we and others have shown that you can record decision-related activity from a number of different areas in the brain, right? So I showed you something from parietal association cortex. We can record similar activity from frontal association cortical areas. from subcortical areas like the basal ganglia, right, or the superior colliculus or so on. So it is really a very distributed process, right? In terms of left versus right, well, in our case, yes, there is a difference, and the difference for that is because we ask them to use eye movements to report their choices, right? So those are being planned as movements that are being performed in a particular direction, and there is a laterality between the two halves of cortex. So responses basically for one side will recruit neurons on one side of the brain, and the responses in the opposite side will recruit neurons on the other side of the brain. Right? Sure.
Hi, yeah, you mentioned that these methods are difficult to implement in humans. There are, transcranial and scalp EEG methods. I was wondering if those could be used for model-based control, or if there are issues with resolution and accuracy and interventional variability.
Right. Ideally, of course, you would want to be able to do this non-invasively, but as you just mentioned, right, so there are strong limitations in terms of, right, spatial and temporal accuracy of those. So right now, we are pursuing the invasive approach, right, but I wouldn't rule out that in the future, hopefully at some point, something similar could be done non-invasively, but we don't know yet.
I was wondering if you'd worked with stimulation in the animal models of choices and found whether or not you could stimulate a certain region and find them making a choice based on that stimulation, or if anything of that sort of deal has been done.
Yeah, we have done stimulation studies. So we haven't done the closed loop stimulation yet that we want to do in this case, but we have done stimulation studies, and again, we and others, and you can definitely systematically influence the decisions, right? You can, for example, strongly bias them, right, to make one decision more than the other and things like that. So you can manipulate the brain activity that the brain in itself uses and reads out to make those decisions. Yes, so that.
Is stimulation of the brain.
Yes, okay, so the question was about, right, if you stimulate the brain, you might actually end up changing the brain, right, and what the consequence of that might be for disease development in the long run. So that is a very interesting question that will have to be addressed at some point. Yes, it is possible that the stimulation could induce plastic changes in the system, right? And so we would certainly want to know what those are, right? So certainly one thing that we've been thinking about is the first thing, can we track those changes, right? So I told you that we learn these kind of dynamic models, and if the system changes, the model wouldn't be appropriate anymore. So can you even keep your model up to date, right, to keep up with those ongoing changes, right? And these are certainly very important questions that we'll have to address at some point, yeah. But we haven't been doing any stimulation for longer periods of time, right? So basically things we've done so far were acute stimulations where you run a session, right, and then you terminate your stimulation again. Yes, of course, exactly, right? But it is an important question, no doubt.
Hi. I was curious if you're looking for less invasive techniques. It's probably really far out there and we're not quite probably at a place where we can do this, but do you think there's any future for optogenetics for less invasive techniques? If you use induced maybe stem cells that can respond to blue light, do you think that would be a decent way in the future to kind of use these models and make it less invasive?
Right. I think you've first suggested optogenetics, right?
Yeah.
So yeah, optogenetics, right, wouldn't necessarily be much less invasive. It could still be an interesting way of stimulating even with more precision than what we can do with electrical stimulation, right? There's still many problems to be solved, right? Optogenetics works really well in rodents when you can genetically modify your animals, right? If you cannot do that, you have to use these AAV viruses that we heard about in the first part to transfect those cells to be able to use optogenetics. And this is already challenging right now in animal models other than rodents. So making that work in humans will certainly be a big challenge, but I wouldn't rule it out that at some point that is possible, or other kinds of non-invasive, right? So for example, ultrasound is getting better as a stimulation technique, even of deeper structures in the brain. So something like that could work as well. So there's many different approaches that are currently being pursued.
Hi. So as I understand, like the model you were showing with like the different axes, they're like a big combination of different neural activities at the same time. So when we're limited in how we're able to like stimulate the brain, are we really able to like navigate the field well enough to be able to control behavior?
So that you have to strike the right balance, right? So you said correctly, right? So usually the space that we are operating in the model is much lower dimensional than the neural activity that we recorded initially, right? So of course what we are doing is during the model identification, we are testing how many of those dimensions we really need to capture, right, a big portion of the ongoing neural activity. And so you have to find the right balance. It has to be high dimensional enough that you can capture that, right? But you also don't want it too high dimensional because it makes just things overcomplicated, right? How well that works for really controlling behavior, I don't know yet, right? So for that, we really have to run those closed loop stimulation experiments that we haven't been able to do yet. So,
last question. All right, thank you so much, Dr. Ditterich.